Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Vanjade.
Today, main topic of the discussion is bisubstrate reactions. Bisubstrate reactions are of two types. First one is sequential or single displacement reactions and ping-pong or double displacement reactions. Sequential or single displacement reactions are of two types, ordered sequential and random sequential reactions.
First of all, what is bisubstrate reaction? The enzyme catalyzed reactions with two substrates and yielding two products is called bisubstrate reactions. Bi means two. In the previous few units, you learned about the enzyme kinetics of single substrate enzyme catalyzed reactions. In this unit, we will discuss about the bi-substrate reactions. Bi-substrate reactions account for about 60% of the known biochemical reactions. Therefore, it is very necessary to consider the kinetics of bi-substrate reactions. So, along with the kinetics of single substrate reaction, we should know about kinetics of bi-substrate reactions. And above that, other multi-substrate reactions are quite complicated. Therefore, we are not going to discuss about that. We will only discuss about bi-substrate reactions. What is Bi-Bi reaction? A reaction catalyzed by a single enzyme in which two substrates and two products are involved. It can be represented as A plus B to P and Q are the products, where A and B are substrates, two substrates, and P and Q are the two products, and E is the enzyme. Bisubstrate reactions involving two substrate, two product, that is Bi-Bi reactions, are generally transfer reactions. Including oxidation and reduction reactions, and we can represent that as AX plus B, that is equal to BX plus A, where X group is transferred from AX complex to BX. So we can represent bisubstrate reaction as A plus B to P and Q product, or we can represent as AX plus B to BX and A as the reaction equation of the reaction. In the first case, A and B are two substrates and P and Q are two products, whereas for second case, here AX is a substrate and B is another substrate, and in that case BX is a product and A is also a product, where single enzyme E is involved.
W. Cleland pioneered the kinetic and mechanistic study of enzymes involving multi-substrate reactions. A systematic classification of mechanisms and the corresponding kinetic equations were probably first time made by him. Let's know the nomenclature system introduced by W. Cleland for representing enzymatic reactions. Substrates are designated by the letters A, B, C, and D in the order that they add to the enzyme, whereas products are designated as P, Q, R, and S in the order they leave the enzyme. Stable enzyme forms are designated as E, F, and G, with E being the free enzyme, if such distinctions can be made. The numbers of reactants and products in a given reaction are specified in order by the terms uni, that is for 1, bi is for 2, ter is for 3, and quad is for 4. Through example, you can understand it. A reaction requiring one substrate and yielding 3 products is designated as uniter reaction, one substrate, three products, unitary reaction. Whereas two substrates yielding two products is designated as Bi-Bi reaction. Similarly, if three substrates, three products, that will be Tate reaction.
Bisubstrate reactions can be divided into two groups, sequential or single displacement reactions and ping-pong or double displacement reactions. Sequential reactions can be ordered or it can be random sequential reactions. I'm going to discuss all of those reactions one by one. First of all, sequential or single displacement reactions. In this type of reaction, all the substrates bind to the enzyme before catalysis takes place to release the product. So in this case, first of all the substrates will bind to enzyme and after that one by one products are released. In that case, a ternary complex of the enzyme and both substrates are formed. There are two types of sequential reactions: ordered sequential reactions and random sequential reactions. First of all, I am going to discuss about ordered sequential reactions. In this type of enzymatic reaction, substrate molecules bind to the enzyme forming a ternary complex. As I told you, in case of sequential reactions, there will be ternary complex formation and then the products are released in a defined order sequential manner. Therefore, it is called order sequential reaction. So, binding and releasing of binding of substrates and releasing of products, there will be a defined order sequential process. So, we can represent, and therefore the order of binding and leaving the enzyme is compulsory, well precise and well specified. Therefore, it is also known as compulsory order mechanism. If you assume that two substrates A and B binds to the enzyme E, the mechanism can be represented as a in this diagram where E is the enzyme. First of all, A will bind to E and in that case AE enzyme with substrate A complex is formed and after that, with AE complex next substrate will bind and we will get AEB ternary complex, then PEQ, and from PEQ first Q will released. Q is the product, first product, and P is the second product, or that sequence can be reversed. That means if substrate B will bind first, after binding substrate B with enzyme, A second second substrate will bind, and in that case A complex is formed. After that QEP, and in that case product P will be released first and then product Q. So if you see these two equations, two series, so you will find that there is a defined, well precise or well specified order of the binding of substrate and releasing of product. If A substrate is bind first, then B will bind second, and in that case Q is the first product release and then P will be released. Or we can represent in another pattern where you can see that here with E first substrate A is bound, then AE complex is formed, after that B substrate is bound and AE ternary complex is formed. from AEB, PEQ complex is formed, and from PEQ, first of all Q product is released, then from PE, then second product is released. This is another view of order sequential reaction. Now I am going to give another plot, which is Cleland plot. A diagrammatical summary of sequences of reactions is also represented in the form of Cleland plot, in which the enzyme is represented as a horizontal line. Here you can see a horizontal line is there, and the arrows, here you can see arrows. There are two downward arrows and two upward arrows. So there is a horizontal line, and the arrows are used to represent the arrival and departure of substrates and products. A and B two substrates, and here Q and P are two products. So these are two signs for arrival, and these are signs for upward arrows for the release of the products or departure of the products. The leading substrate A binds first, followed by B. A reaction between A and B occurs in the ternary complex AEB and is usually followed by an ordered release of products P and Q, or release of products Q and P. First Q product is released, then P product is released. So there is a sequence here, or we can write in this way also, where first substrate is B and second substrate is A, and first product is P and second product release is Q. So this is the these are two Cleland plots for the ordered sequential reaction, bi-substrate reaction. We can also represent ordered sequential reaction in this way, where structure of E is this, and with enzyme E, A substrate is bound first, and after binding with substrate A, there will be slight change in the conformation of enzyme. So if you carefully see the structure for the these two active sites, you will see that after binding with substrate A, there is the changes in the structure of second active site. So after conformational change, binding of substrate B will be there. If you compare active sites here and active sites there, we'll find that this substrate B can't be bound to enzyme, because the conformation is completely different. Active site is not suitable for B. Here it can't be fitted properly, but after binding with A, the conformation of E is changed, and you can clearly see that the structure of this active site is well fitted with the substrate B, and then only substrate B will bind to the EA complex, and AEB ternary complex is formed, and after that two products Q and P are released sequentially, first Q and then P. So in this case, in ordered sequential pathway, first substrate causes a conformational change in the enzyme. You can, I already discussed that. This is clearly shown from these two figures, where after binding with A substrate, there is a conformational change in EA complex. Therefore, after that, substrate B will be bound, and AEB ternary complex is formed. Now, I'm going to give you some examples of ordered sequential reaction. Some of the well-known examples include reactions catalyzed by the enzymes such as citrate synthase, lactate dehydrogenase, and aspartate transcarboamylase. So these are three enzymes. These three enzymes catalyzed reactions are examples of ordered sequential reactions. Or other examples are ATP sensitive K channels and H, K pumps, etc. are also some of the other examples of compulsory or ordered sequential reactions. Now, random sequential reactions. There is no specific order for substrate binding. Any substrate can bind first to the enzyme and any product may be released. So there will be no order. Therefore, it is called random sequential reactions. There is no specific order for substrate binding and product releasing. All the possible binary enzyme substrate and enzyme product complexes are formed rapidly and reversibly when enzyme is added to the reaction mixture. In the reaction mixture, there are A and B substrates and P and Q are two products. This is the random sequential reaction mechanism, where we can see you can see that the A and B are two substrates. There will be no order for the binding with the enzyme. AEB, the enzyme substrate binary complex, ternary complex, and PEQ are substrate products ternary complex, and Q and P are the products. So there is no order for the substrate binding or product releasing. We can represent random sequential reactions in the Cleland plot. This is the Cleland plot for random sequential reaction mechanism. You can see clearly that these are two arable sites of substrates. Here either A or either B, and after that either P first release or Q. These P and Q are two products, so there will be no order in the binding of substrates or releasing of products. Let's see examples of random sequential reaction. Random sequential mechanism is occurred typically in multi-enzyme complexes like hemoglobin, ligand operated channels, and tyrosine kinase receptors. All cells native to the central nervous system, including neurons, express creatine kinase, that is CK, the enzyme that catalyzes the equilibrium among phosphocreatine, ADP, creatine, and ATP. It is very essential enzyme in our body. Creatine kinase or CK mediated conversion of creatine and ATP to creatine phosphate or phosphocreatine and ADP is an example of random sequential reaction. Here you can see that ATP and creatine are two substrates. So binding of ATP and creatine with enzyme creatine kinase is random. In that case, ATP is creatine ternary complex is formed, and after that ADPE creatine phosphate binary ternary complex is formed. After that two products ADP and creatine phosphate are released in random manner, and there will be, after that there will be free enzyme. And another diagram of random sequential reaction is this, where we can see that this is the structure of enzyme. Either A bind first or B, and AEB ternary complex is formed, and Q and P are the two products. You can clearly see that there is no change in the conformation here after binding with substrates, whereas in case of order sequential reactions there was conformational change in the enzyme structure. So these are two main difference between random and order reaction. In order reaction there is conformational change in enzyme after binding with the first substrate, but in case of random sequential reaction there is no change in the conformation of enzyme if substrates are bound. Let's know about Ping-Pong Bi-Bi reactions or non-sequential reactions, or it is also known as double displacement reactions. Group transfer reactions in which one or more products are released before all the substrates bind the enzyme are called non-sequential reaction. There is no sequence in the binding and releasing of products. The main feature of double displacement reactions is the existence of substituted enzyme intermediate in which the enzyme is temporarily modified. I have also told that in case of order sequential reactions there was some conformational change. In case of Ping-Pong Bi-Bi reactions, similarly enzyme intermediate there will be temporarily modification in the enzyme. In this reaction no ternary complex is formed. So main difference between the sequential and non-sequential reactions is, in case of sequential reaction there is ternary complex formation, but in case of non-sequential reactions there is no ternary complex formation is occurred. Ping-Pong Bi-Bi reactions are two-stage reaction. First stage that is ping. In that case a functional group from first substrate A is transferred to the first product here, forming a temporarily modified enzyme E prime. In that case you can see that E is bound to first substrate A, and AE binary complex is formed. After that PE prime, and from PE prime P product is released first, and in that case some changes in temporarily modification will be there in the enzyme, and E prime enzyme is formed. After that, in the second stage, that is called the POM, the functional group is displaced from the temporarily modified enzyme by the second substrate, that is substrate B, yielding second product, that is product Q, and regenerating the original form of the enzyme. So in this case first substrate A is bound, and after that first product is released. After that, when first product is released, there will be some temporary modification of enzyme, and after that, with that modified enzyme, second substrate is bound, and then second product will be released. Let's see the schematic diagram of Ping-Pong Bi-Bi reactions. You can see here, this is the structure for enzyme. First of all, there is a single active site first. With that active site, substrate A is bound and AE complex is formed. From AE complex, first product P is released, and after releasing the product, there will be some changes in the conformation of E. Therefore, it is transformed to E prime. You can see here the active site structure is changed. You can see the active site structure is changed. It was like in this way, but after first product release, the conformation is changed to this type. And only after that change, substrate B will be fitted properly with the modified enzyme, and BE prime complex is formed from BE prime. Second product Q is released, leaving the enzyme in the initial conformation. In this type of reaction mechanism, enzyme has only one binding site. You can see only one binding site is there. Since only one substrate binds at a time, first A and then substrate B, there is no room for the second substrate. Whereas we found that in case of sequential reactions, there was two active sites for two substrates. In case of Ping-Pong Bi-Bi reactions, there is no room for the second substrate. Only one active site is present. The release of first product exposes the binding site on the enzyme to the second substrate. Let's see the Cleland plot for Ping-Pong Bi-Bi enzyme catalyzed reaction. You see, this is the horizontal line, and first A substrate is bound to enzyme, and then product P is released. After that, there will be change in conformation of the enzyme. With the modified enzyme, B substrate is bound. This downward arrow means the substrate binding, and after that the Q product is released. The upward sign is for product release. This is the Cleland plot for Ping-Pong Bi-Bi enzyme catalyzed reaction. Example of the Ping-Pong Bi-Bi reaction is transfer of amino group from aspartate to alpha ketoglutarate by the enzyme aspartate aminotransferase. In this reaction, aspartate and alpha ketoglutarate are two substrates where amino group of aspartate is transferred to alpha ketoglutarate, forming glutamate and oxaloacetate as the products. In this case, enzyme is aspartate aminotransferase. This is the Cleland plot for this reaction, where you can see that E is AST, that is aspartate aminotransferase. First substrate aspartate is bound to enzyme, and enzyme aspartate complex is formed. After that, E prime oxaloacetate binary complex is formed, and from that binary complex oxaloacetate is released. This upward sign is for product release, and after first product release, there will be some modification in the enzyme, therefore it is transferred to E prime, and with E prime second substrate, that is alpha ketoglutarate, is bound, and then second product glutamate is released, leaving the enzyme in the original form. This is the Cleland plot for the AST catalyzed reaction. Let's see at a glance three types of bisubstrate reactions. First of all, order sequential reactions. This is the schematic diagram for order sequential reactions. Then random sequential reactions, and finally ping-pong or double displacement reactions. These are three schematic diagrams. From these three diagrams, you can easily compare their difference. In case of ordered and random sequential reactions, you can see that first of all substrate binding is completed. After that, products are released. Whereas in case of ping-pong or double displacement reactions, first substrate is bound, then first product is released. After first product release, then second substrate is bound, and then second product is released. So binding and binding, then first substrate binding, then first product release, then second substrate binding, and then second product released in case of ping-pong or double displacement reactions. And another difference is that in case of order sequential reactions, there was conformational change in the enzyme, and similarly in case of ping-pong or double displacement reaction also there is a change in the conformation in case of enzyme, but in case of random sequential reactions there is no change in the conformation after binding with the substrates. In case of order sequential or random sequential reactions, there are two active sites for two substrates, whereas for ping-pong or double displacement reactions there is only one active site for two substrates. After binding with first substrate and releasing of first product, there will be change in the conformation, then only second substrate can bind. I hope you understood the topic very well. Thank you very much for watching this video.